Our seminar speaker is Dr. Anthony Burnetti. We call him Tony, who's a senior research scientist in the School of Biological Sciences here at Georgia Tech. He's an astrobiologist whose research focuses on the evolution of multicellularity, and particularly using lab evolution and synthetic biology in yeast, which he's going to be talking about today. And then more recently, I know he started some new direction with research, looking at major evolutionary transitions via theory and also directed evolution experiments. He has a bachelor's in cell biology and minor in astronomy. So he's been interested in astro bio for a long time from the University of Maryland, and a PhD in genetics and genomics from Duke University. And then since 2017, late 2017, he has been here at Georgia Tech, first as a postdoc, then a research scientist, and now as a senior research scientist. He's a very active member of our astrobiology community. I love his talks. I always learn something new, and so I'm really happy that we get to see an update on your research. And so thank you for talking with us.
Right. Let me make sure that the view here is going good. Is it possible to get this bottom bar out of the way? Okay,
No.
Whatever. All right, cool. So thanks for that intro. So like you said, I mostly do research on the evolution of multicellularity, but that is not the only interesting major transition in evolution that occurred on Earth. So today, I'll be talking about the evolution of phototrophy and the way that apparently priority effects have inhibited its repeated evolution. And this talk is largely based on a paper which, after a couple of years of improvement, has finally been published as of this Monday. So there we go. Oh, this is thrilling. So, here we go. So the big question, I think, that I'll be talking about here is, are major evolutionary innovations and major evolutionary transitions predictable or contingent? And everyone asks this question, no one gets the same answer. You will find people saying wildly different things about the history of life on Earth and whether evolution is just some kind of pure chance. You can't predict what's going to happen, or, well, all these steps make some sort of sense, and this is just obviously what happens. And the interesting thing about that is everyone's looking at the same thing, and they're coming to these wildly different conclusions. So what this tells me is it's all vibes. No one's being systematic about this. So I would like to try to be systematic about this in some way, and I think I found a little bit of a way to do so. So, point is, Earth has changed a lot over the billions of years. The planet has transformed and life has innovated. You went from small anoxic biospheres to big microbial biospheres to even bigger biospheres today with lots and lots of big complicated things. And it's full of major innovations. Some major innovations, like multicellularity, keep happening. Like everyone talks about, there's about 5 complicated origins of multicellularity, but if you look closer at any form of multicellularity, there's 50 or 60 that people can agree is real multicellularity. So apparently, this major evolutionary innovation is easy. It keeps happening. And people largely agree on that. But there's a lot of major innovations that are what people call evolutionary singularities. These are things that appeared exactly once and they changed the world. Deepest, biggest one would be something like, say, the origin of translation in the genetic code. We got some people here at Georgia Tech studying that, and it's very important, the ability for there to be coded protein at all. Things like the origin of eukaryotes, that seemingly happened once, and a lot of the major things that have happened in the last billion years are the result of that. And you also got things like particular metabolic pathways. The classic one I like to talk about is nitrogen fixation. There is one enzyme on Earth that will take nitrogen gas from the air and turn it into biologically available protein mass. That's really important, and it apparently only happened once. So these things that happen once, why might an evolutionary innovation be singular? Because when it's singular, it's difficult to study. You only have one example. You can't compare it to anything. So you could say that maybe they're just extremely unlikely to evolve. And we see these in the particular history of our planet because of anthropic selection effects. We're here, so we see things where we see a world where they happened. You could also imagine that maybe there were multiple origins and you just have progressive extinction of different lineages over time, attrition. Or, and this is what I think is interesting, this could be the result of historical ecological processes that limit what you can see, specifically evolutionary priority effects. A priority effect is, in ecology, is when, say you have a bare field and something comes and colonizes it, and it prevents something else from coming in and colonizing it because it's well established. You can't unseat an incumbent. An evolutionary priority effect is where this occurs rather than in colonization from outside, evolution from a pre-existing state where something didn't previously exist. So something appears and it prevents the evolution of anything too similar to itself. So I will be examining this idea of evolutionary singularities and priority effects by looking at a very important innovation, and that's phototrophy. Phototrophy is the ability to use light for biological energy. And this drives just about all biomass on Earth. There is non-phototrophic primary production on Earth. It's tiny. We're literally talking a millionth the size of the primary production of photosynthesis on Earth today. I have this little bar graph here on a log scale of trillions of moles of carbon that can be fixed by oxygenic photosynthesis and oxygenic photosynthesis. The black bar is if literally every geologically available source of carbon fixation was 100% tapped, and that's not happening. So the phototrophy appeared and it completely changed the world. There's a reason that the oldest evidence of life on Earth that no one argues about is evidence for photosynthesis. It changes everything. It's the reason that you can tell Earth is alive from across the galaxy with a good telescope versus maybe not being able to tell it's alive if you were standing on it. So it's very important. I think it's the second most important thing in the history of life after the ribosome itself. But the interesting thing is, phototrophy was invented twice, exactly twice. Not once, and not a bunch of times. And that is really weird. So these are just two pictures. On the right, moss, seaweed, something like that. You're familiar with that. That is chlorophototrophy. Everything green you've seen outside is a subset of chlorophototrophy. But there's a separate origin that you mostly find in microbes called retinalophototrophy. You're most likely to physically see it if you're like flying over a salt flat where you have all those fancy archaea in those really salty waters that are full of this machinery. So you've got two origins. 2 is a weird number. I don't remember who said this originally, but some physicists like to say there are 3 natural numbers, 0, 1, and lots. Because either something can't happen, it happens all the time, or there's some reason that there is some special instance of something. 2 is weird. 2 means something. And I think that this can tell us something. So let's talk about these 2 origins of phototrophy. The one that we're all most familiar with are the chlorophototrophic reaction centers. Oxygenic photosynthesis uses this, and a bunch of other bacterial groups. It's actually super diverse. And it's a fairly big and complicated protein system. You've got dozens of chlorophyll or bacterial chlorophyll molecules in a big complex that's part of an electron transport chain. They can be up to 350 kilodaltons in size. And these use photochemistry to move around electrons. An excited chlorophyll moves an electron from one molecule to another, and then it gets fed into an electron transport chain, and you pump 2 to 4 high voltage protons around. Or you can take that high energy electron and use it to reduce carbon or nitrogen for carbon fixation and nitrogen fixation. And this is big and complicated. The smallest of these systems, you need about 30 genes. It does horizontally transfer around, not often. You can point to a tree of the bacteria and like point to the times that it has. And this is like the simplest version of it on the left here. It can get connected to a whole bunch of other doodads. You can have all kinds of antennas attached to it, or in the case of oxygenic photosynthesis, some extra proteins around that sort of stabilize some fancy ions that let it rip apart water and make oxygen. And so in all these cases, you're taking an electron from somewhere out in the environment, boosting it up and doing it for carbon fixation, or you're running an electron transport chain in a circle, making energy. And I want to point out, everything that uses chlorophyll or bacterial chlorophyll, it's got a single common ancestry, all the oxygenic and anoxygenic. It all goes back to one ancestral homodimer protein that's then diversified and changed and had stuff added to it over time. And it's all based on a modification of heme. You can tell this is all downstream of like other redox chemistry that things do. And it's all wired up to electron transport chains. And I just want to point out this, the relationship between oxygenic and anoxygenic photosynthesis. Some people like to assume that oxygenic photosynthesis is much more complicated and came much later. It's actually not that clear. The split between them is really deep and complicated, and people argue about it a lot more than you might think looking around in the older literature. So that's chlorophyll. What about rhodopsins? So retinalophototrophy uses a single, about 27 kilodalton protein called a microbial or type 1 rhodopsin. It's just a little membrane pore with one pigment molecule stuck in the middle of it. And it pumps 1 proton per photon it absorbs by just changing shape and forcing a proton across the membrane. And it captures a lot less of the energy per photon. It captures less than 1/10 of the energy of each photon that falls on it. But man, is it simple. The protein itself is just one little gene, and if you want to make the pigment, you just add another 1 to 4 genes on top of that, and you've got and you can horizontally transfer this, and it horizontally transfers like mad. It's all over. Unlike in chlorophototrophy, where you can tell it definitely happened in the bacteria, no one can tell where this happened. It's everywhere. And I've even stuck it into my yeast and they turn purple. It's cool. So these things are very different. There's chlorophyll uses redox chemistry versus the other one using a proton pump. They're complex and large versus small and simple. And it can conserve a large amount of the photon's energy or a small amount of the photon's energy. They're very different. So let's try to analytically compare these two. What can we learn by comparing them? So I've done some work where I've done some physiological modeling of these metabolisms. I've made a model of their energy capture, and I'm mathematically comparing their capabilities. So I think that we can tell something interesting by looking at the relationship between the two halves of this unique, interesting dual singularity, and maybe learn something about the true singularities here. And this is all downstream of a paper that was published in its final form, finally, this Monday. So go take a look at it if you want sometime. So let's take a look at the two different systems physiologically. So on the left here, you've got one of the well-studied but simpler chlorophyll systems. You've got sort of the central engine of the thing with some pigment molecules inside it, and then a bunch of antenna pigments around it that can gather light and dump the energy into it. On the right, you've got a rhodopsin, just one little protein. And it turns out a lot of these have been studied very well. So what do we get? I took a couple of these systems, 2 chlorophyll using systems and 2 rhodopsin using systems, and I just did some math and I figured out some properties of them. So this is what you get when you talk about the maximum energy flux per unit protein. If you have a gram of the protein, how much energy throughput will it have? Or protons per second per kilodalton is the units that was useful here. Turns out oxygenic reaction centers and oxygenic reaction centers pretty low, and that's because they're big. They conserve a lot of energy, but they are huge. Whereas the rhodopsins, depending on the exact system and mostly how fast they can run, they can have really high energy fluxes when they are running full tilt in a bright light. And the other interesting thing is the chlorophyll, there's lots of pigment molecules. They've got a big cross section, a big light funnel, funneling in all that energy, whereas the rhodopsin just has this one little tiny dye molecule. So they actually behave very differently at different light intensities. This is how they work at different light intensities. The chlorophyll using ones, they do really good at low light, but then they saturate and they stop doing better as the light increases. The right side here, the very right side is about direct sunlight at the equator. Whereas the rhodopsins, they just keep doing better as you add more light. And you add more light, they just keep doing more and more and more and more. So that's really interesting. The chlorophyll does way better at very low light, but at high light, the rhodopsin actually does better in terms of the amount of energy you get from an investment in the protein. But then, flux per unit protein isn't the only thing. How much of the light energy falling on you do you capture? So this, again, is the maximum. It turns out you get the opposite pattern. The chlorophyll using systems, especially the oxygenic ones, mostly because they have, again, the huge antennas attached to them, can get lots of energy per unit light that is falling on them, whereas the rhodopsins have less. And again, this is the maximum they can manage. If we look at it at different light levels, once again, the chlorophyll wins handily at the low light, but then it falls off as the light gets more and more intense. Whereas the rhodopsins, they're doing a little bit less well, but as the light density increases, it's still capturing a fairly similar amount of the energy that's falling onto it. So that's really interesting. There seems to be a trade-off here between the flux per unit infrastructure that you've invested and the yield per unit resource that's around. And it turns out that that trade-off is actually all over microbial metabolism. You find it in lots and lots of metabolic pathways for some deep thermodynamic reasons I won't get into. But so that was looking at very particular systems that are around today, though. The thing is, those are particular systems optimized for a particular environment. What if we look at their theoretical performance in every environment? And what do I mean by environment? I basically mean light intensity. That's within the scope of my ability to model here. So here you've got these systems. You've got this central engine with some pigments and various attached antennas dumping energy into that central engine. I created a simple model of it that's just very chunked. I just have a central engine with a mass and an amount of ability to gather light that runs at a certain maximum velocity and has a certain yield per photon that absorbs it, or that is absorbed. And then I have a variable amount of antenna pigment attached that can absorb a certain amount of light. And then that whole thing is recycled with R there, or destroyed by bad interactions with light, because things can get bleached, it happens. And all of this is exposed to different light intensities. And so I'm just abstracting away all the fancy complications into this very simple abstract model. And it turns out you can do some math, you can figure out sort of the flux per unit protein and the amount of energy per unit light of these things by just doing the math on exactly how much of this protein will be functional versus bleached, and the rate at which it will be absorbing light and how quickly it will be running. And so basically, what I did was I took each of these systems, I gathered the relevant variables from the literature for a particular central engine of a particular chlorophototrope and a particular rhodopsin. And I allow the light to vary from 0 to direct sunlight at the equator, and I calculate at each light intensity the optimal amount of antenna where it does the best, where these fluxes and yields are maximized. Because if you have too much antenna, then your central engine is saturated. If you don't have enough, it's not running at its full capacity. So there's an optimal amount of antenna. So I figured out an optimal chlorophototrope and an optimal retinalophototrope at every light intensity from 0 to the maximum that anything on Earth sees. And I see some interesting things. So here you have log scale light intensity on the bottom, and here you have the efficiency per unit light, how much of the light energy is gathered by the system on the left, and the flux per unit protein, the sort of wattage of the machinery on the right. And you can see there's a crossover point. At lower light intensities, chlorophyll wins hands down at both things. At higher light intensities, rhodopsin wins hands down at all things. And something interesting, if you actually plot these two variables against each other, you see something interesting. Here you have the flux per unit protein versus the yield per unit resource. And this represents what you call a Pareto front. Everything actually alive will probably exist somewhere on this combined front, because anything that's back here because it doesn't have enough, or it has too much antenna, or it's using the wrong kind of system, is not optimal and will evolve towards the front, and then it is forced to sort of evolve along the front. That front should represent everything that's actually alive. And this is interesting. You see that there's sort of two regimes. You have the high light regime where the rhodopsin wins, and the low light regime where the chlorophyll wins, at least in terms of energy production. And when we look at the properties of these systems, I can tell you the optimal amount of antenna in all these things. It actually corresponds with real world antennas. At low light intensity, you get arbitrarily big antennas, which is true. You get some weird photosynthetic bacteria that have organelle-sized crystals of chlorophyll that can be just significant fractions of the mass of the cell. But then you get sort of at the crossover point, you have a certain mass of antenna. This crossover point is actually within about 20% of the mass of the smallest antenna that anything on Earth has with chlorophyll. So I think I'm onto something. And and you get very small optimal antennas, like way smaller than any actual proteins for the rhodopsins, and you don't actually see antennas on the rhodopsins. So I think that this fits with what you see in reality. And also, it also corresponds with where you find these different systems in the world. So this is data from a survey of the ocean. Looking at a bunch of different properties of different samples from different depths all along. Somebody was doing a transect of the ocean. So I just plotted the amount of available light and the amount of chlorophyll and the amount of retinal that was there. And the dotted line is that crossover point. So you can see above that crossover point, you got a lot of rhodopsin. Below that crossover point, you got a lot of chlorophyll. I think yeah.
First of all, this is amazing. Second of all, could you just clarify what like the small antennas that are smaller than the protein?
Yeah. So
clarify that.
So you got so in the chlorophototropic reaction centers, you have sort of this little dimer at the center, which is the actual engine of like electron transfer, and then the smallest antennas that you get are what are called integral antennas, just little antennas that are physically part of the same peptide that are off on the sides of it, and they just contain a bunch of chlorophyll, and they just will absorb light and dump their energy into that central engine. And then
What are the antennas for the retinol?
The retinal doesn't actually have protein antennas with additional pigments. No one's found one with an antenna like that. What they have found is one that has a little carotenoid wedged into a crack in the protein that just slightly increases its absorption cross section.
And that changes as you get to higher light.
So what I find is that as you go to higher light intensities, the optimal antenna falls all the way, would fall all the way to 0 for chlorophyll, falls very small. We're talking like 2 kilodaltons at bright light for the rhodopsin. At the crossover point, you get that there would be an optimal antenna of like a couple dozen kilodaltons, but that's only at the very low end of the range, and you don't actually see it. You don't actually see that anywhere. I don't think, and given the whole range of rhodopsins, I don't think it's strange that you don't see any antennas attached to rhodopsins.
I see. Okay.
So
got it.
Yeah, the only antennas you see attached to rhodopsins are some of them have a carotenoid literally shoved into a crack between 2 helixes in it, absorbing a little bit more light. Yeah.
And so the equation for FP and FL, is it output upon input? Is it output upon input? Because I see the equation based on how it's built, L into Vmax into AX plus B. Is it output upon input or?
I'm not sure what you mean. The Vmax refers to the maximum speed at which the central engine can run.
Okay, but the equation is output upon input, like what is FP?
It's the flux per unit protein, the protons pumped per kilodalton per second of the full machinery at a given light intensity for all these variables of the system.
So can you give the intuition just behind how you derive that equation?
Can we leave the?
Yeah, we'll get to that in a minute. We'll come back to that. So yeah. So any case, what's really cool here is I think I found that there's this really nice precise niche partitioning between the 2 existing forms of phototrophy. They divide the world up into sort of a low light, high yield, low flux specialist niche and a sort of a high light, low yield, high flux generalist niche. And I think they'd be unlikely to divide this niche so precisely if the properties were random. I think that this suggests that this is the result of evolutionary priority effects. Each of them has suppressed the origin of anything too similar to itself. And you have instead allowed the existence of something that is different enough to fill the rest of the niche. And so it would look something like this. You've got that Pareto front. When things originate, they suck. Whenever they first are able to interact with light, they're bad on both axes. But they will adapt, they'll get better, they eat their free lunches, but then they get to the Pareto front and they're forced to diversify along it. And each of them is filling a given part of the Pareto front, and together they are suppressing everything else. But you might ask, what would have happened when the very first one came into being? Wouldn't it have filled the entire front and prevented the evolution of the second one? Yes, you would think that, and I think that actually tells you that retinalophototrophy came first. And that's because chlorophototrophy doesn't just make energy, it can also fix carbon. Retinalophototrophy, it doesn't do any redox reactions, can't make any NADPH to fix carbon. It also doesn't pump the membrane up to a high enough voltage to do an alternate process called reverse electron flow. So it just can't fix carbon. So I think that chlorophototrophy could break through a retinalophototroph priority effect, but not vice versa. So because even if it's bad at getting energy, in a world where you are stuck with geologically provisioned hydrogen, the tiniest little bit of light-driven carbon fixation, you win. So I think that this shows that retinalophototrophy probably came first. And that fits with thinking about the 2 systems. Retinalophototrophy, it's just a proton pump. All that you need for that to be useful is that you've got an energized lipid membrane. Whereas chlorophototrophy, those things are plugged into fully functional electron transport chains with cytochromes and quinones and all that good stuff. So the one that's simpler and has fewer prerequisites seems to have come first. Makes sense. And I just want to note, this is me talking about the evidence that maybe you're suppressing the origin of anything too similar to each other. Is there any evidence of suppressing other paths to phototrophy? I think there is. It turns out there's some enzymes that use light in the world. You got photolyase that fixes certain mutations, and you've got photodecarboxylases that turn fatty acids into oils, basically. And they all use light to sort of push an electron around in a little circle in their active site just to rearrange some bonds. They've got pigments. They've got a flavin, a redox cofactor, which just happens to interact with light, that gets excited and pushes an electron around in its excited state. And then it's got folate, freaking vitamin, as an antenna pigment, which also, because of the way it has to move around electrons, just happens to interact with light. And this looks just like what you think like the pre-chlorophototrophy might be, except with heme instead of, say, flavins, and where you would then, say, optimize it into a real phototrophic pigment. So if you took this and you tried to optimize it to drive a metabolism, I think this is exactly the sort of thing that would be right at the base of chlorophototrophy. So, in summary, this is what I think happened. I think you sort of invented retinal phototrophy. It spread to a bunch of niches. You eventually invent chlorophototrophy. It spreads. They smash into each other. They partition the world between each other. And ever since, they've been suppressing the origin of other phototrophic systems. So that's interesting. This dual singularity, because you can actually compare them, you can see evidence of these priority effects. And of note, some people that I've talked to about this think this resembles something called the purple Earth hypothesis. There's some people who have looked at these two systems and think that since the retinal is so much simpler, and for also just reasons of the shape of their absorption spectra, they think that retinal could have come first, and the Earth could have been. You could have had purple Earth, and this could be a signature of early biosphere. Should we be looking for this instead of the chlorophyll red edge out in the universe? I actually don't think so. I think that because apparently the limiting factor for these things is their interaction with each other, I think the time between them could be possibly very short. And you could even have a very short time from the origin of life itself to these things. So I think that this idea doesn't actually necessarily support this idea terribly strongly. So this has important implications. These two systems partition ecological niche space. Together they're engendering this evolutionary priority effect. Retinal phototrophy probably came first, and the time between them and the time to the origin of life from them could be potentially very short if this is the limiting factor, rather than them being difficult to evolve. And there are other possibilities, though, for other major evolutionary innovations. This is what we seem to see for phototrophy. You don't seem to see priority effects, or there's just a huge number of niches for something like multicellularity. So apparently some major innovations don't encounter this sort of thing, but you could also imagine a circumstance where the ecological niche space is just simple, and whoever comes first wins and dominates forevermore. That you engender an evolutionary priority effect, and you make something that's easy to invent, nonetheless only ever happens once. So what other singularities might also be easier than they look? I have some thoughts. So the origin of life or the origin of the genetic code, things like that. This is a very strong candidate for it being a priority effect. People have thought about this since Darwin. Darwin himself was like to speculate that the transition from non-living matter to living matter, you don't see it happening today because anything that's kind of alive will just get eaten, because it's not very good at being alive by comparison to everything that's around today. Nitrogen fixation or other core metabolisms. I think this is also a good candidate. So you've got this single origin of biological fixation of nitrogen. You've got a class of related enzymes, some of which make a big complicated ion cluster and some of which use a big complicated ion cluster in order to fix the nitrogen. Again, if you have an enzyme that does something very difficult and it optimizes to do it well, how is something that does that same job poorly going to compete with it, especially in a world of microbial horizontal gene transfer? You know, something's going to grab this and use this. Interestingly, the enzymes that are used for nitrogen fixation are closely related to some of the ones that build chlorophyll. I wonder if this is a coincidence or not. But I don't know, it seems interesting. And then there's eukaryogenesis, the big elephant in the room. So you get this single origin of complex cell architecture from the Asgard archaea, which already are doing some slightly interesting things. You've got endosymbiosis and all kinds of novelties on top of these already interesting archaea. Is this a good candidate for evolutionary priority effects? Maybe, possibly. The interesting thing about eukaryotes is they all have dead simple metabolisms. They compete on their interesting shapes, basically. Maybe once you're good at changing shape, it's hard for someone else to invent that ability. However, there's also eukaryogenesis as a singularity. I say hold your horses. There's some interesting other things starting to come out in the literature. You've got some really interesting prokaryotes that are being found. My favorites are Thiomargarita magnifica, this freaking centimeter-long bacteria with very poorly characterized internal membranes that someone needs to do a lot more work on, and genome assemblies that really need to be done with some long-read sequencing, and planktomycetes. There's a video now of a planktomycete bacteria catching, engulfing, and eating an E. coli. These things happen, and I think those would be very interesting for people to look at. I also think eukaryogenesis might also be an interesting thing. I think that everybody loves to say, oh, eukaryotes are so weird, all these things changed at once. I think it might actually be sort of a restricted space of possibilities that I'm actually vaguely pursuing right now, a model of sort of the evolution of genome structure and sort of recombination rates, right? And I think there might actually only be two stable states for your genome structure. I think a couple of variables might have to change together. I think you might need a compact genome, non-repetitive, details that you occasionally recombine with anyone and you tend to delete genes versus all the opposites of those. I think those two might actually only be the stable states. Put a bookmark here. I'm trying to figure out if this actually works. So it might actually be that eukaryogenesis is not as weird as you think, that it's just like one of the only two possibilities. So I can't wait to see if that works out. So in conclusion, for all of this, I think that chlorophototrophy and retinalophototrophy, by looking at this dual singularity, you can see a priority effect that otherwise would be hard to see in a true unitary singularity. I think that the evolution of phototrophy is easier than it looks, and you can tell this because you can actually compare the two of them. I think that a lot of other singular innovations may also be constrained by priority effects and be easier than they look. And astrobiologically, I think a lot of models where chance constrains novelty and where there's anthropic selection effects and where there's a famous one called the hard steps model, where people try to figure out the timing of major innovations in the history of life on Earth as sort of being a random walk. I think these are less favored than a lot of people think, because I think a lot of things that look hard aren't, or at least aren't as hard as they seem. So that's it, what I've got today. I want to thank the whole Ratcliffe team for letting me go off on this in my spare time from pipetting yeast around, and I'll take any questions.